Okay, so welcome back to this next video in which we are discussing transcription factors. Okay, right, so we're in the process of discussing why it is that transcription factors bind to the major groove of DNA. Okay, right, so we are discussing the base pairs of cytosine organic bases with guanine organic bases. So I've drawn a cytosine organic base next to a guanine organic base here. Now what we want to discuss is what's actually going to hold these two together. And by the way, there should be a hydrogen coming off that nitrogen there. Okay? Right, so the two organic bases are going to be held together by a number of hydrogen bonds. Okay, so let me just remind you of how hydrogen bonds, what they consist of. So, basically, to form a hydrogen bond, what you need is a hydrogen atom like this, okay, bound to an atom that is far more electronegative than it. And I'll just remind you of what electronegativity means. So let's draw this bond between this nitrogen and this hydrogen atom bigger down here. Okay, so it consists of two electrons. One electron is from the nitrogen atom, we'll say this one in pink here, this comes from the nitrogen atom, and the other electron here comes from the hydrogen, and that one's in blue. Okay, now, the question is, these two electrons that are in this single covalent bond, and which are being shared by the nitrogen atom and the hydrogen atom, do they sit right in the middle between the nitrogen atom and the hydrogen atom, or do they sit tilted to one side? Well, to answer this question, we need to think about what forces are going to be acting on these electrons here. So, these two electrons in this covalent bond are going to be feeling attraction to positively charged particles because they themselves are negatively charged, okay, and opposite charges attract one another, okay. So, where are the positive charges? Well, the protons which are in the nuclei of the nitrogen and the hydrogen atom, those are positively charged, okay? Remember, nuclei consist of protons and neutrons. The neutrons are neutral and the protons are positively charged, so nuclei overall are positively charged. So both of these electrons here are going to be feeling attraction towards the hydrogen nucleus and towards the nitrogen nucleus. Now the question is, which of those two attractions is stronger? Okay, well, you might say surely it's the nitrogen atom because the hydrogen atom only has a single proton at the center of its nucleus, whereas the nitrogen atom has far more protons. Okay, and therefore you're going to get a greater attractive force towards the nitrogen nucleus. However, it's slightly more complicated than that because you also have to take into account the electrons that are orbiting the nitrogen nucleus and that are closer to the nucleus than these electrons here, and which therefore are shielding these electrons from that positively charged nucleus with their negative charge. Okay, and the hydrogen atom doesn't have any of those because remember, this is the only electron of the hydrogen atom, and this one it's sharing now. Okay, oh sorry, this is the only electron of the hydrogen atom, and this one it's sharing here. Okay, so there aren't any electrons shielding the positive charge of the hydrogen nucleus from these electrons. Okay, so overall it's quite complex to work out how much force these electrons will feel towards each of these two nuclei. Okay, but this is what the word electronegativity means. Electronegativity means how much does the atom attract these electrons in the covalent bond. So this takes into account both the attraction towards the protons and the shielding by the electrons that orbit that nucleus. Okay, so electronegativity means how strongly will an atom pull the electrons in a covalent bond towards it. Now, nitrogen has a much greater electronegativity than hydrogen, so nitrogen will pull on these electrons harder than the hydrogen atom. Okay, so the electrons will sit towards the nitrogen atom, and that will make the nitrogen atom partially negatively charged, whilst the hydrogen atom, which loses the electrons, will become partially positively charged. Okay, this is the first thing you need in order to form a hydrogen bond. You need a partially positively charged hydrogen atom. Okay, and this is known as the hydrogen bond donor. Okay, so the hydrogen with the partial positive charge is called the hydrogen bond donor. Okay, right. Then the other thing you need is a hydrogen bond acceptor. So to form a hydrogen bond, you need two things: a hydrogen bond donor, which is a partially positively charged hydrogen attached to an atom which has a greater electronegativity, which has produced this partial positive charge, and you also need a hydrogen bond acceptor. Okay, now an example of a hydrogen bond acceptor would, for instance, be this oxygen atom here. Okay, bound to a carbon atom like so, because this oxygen atom will have two lone pairs of electrons. Okay, like so. So you need an atom which firstly is partially negatively charged and also which has lone pairs of electrons. So in these covalent bonds, these two covalent bonds between this oxygen atom and this carbon atom, you will overall have 4 electrons, 2 in each of them. Okay, and these will be pulled towards the oxygen atom because oxygen has a greater electronegativity than carbon, so that will make the oxygen partially negatively charged. Okay, in addition, you have this lone pair of electrons here, which is actually a center of negative charge. Then what you can do is you can form a hydrogen bond, which is an electrostatic interaction between the partially positively charged hydrogen and firstly the partially negatively charged oxygen here, but also it's specifically localizing with this lone pair of electrons, which is a center of negative charge. Okay, so this is a lone pair of electrons, and this is just functioning as a center of negative charge, which that partially positively charged hydrogen is now going to associate with. So this bond that we formed here is a hydrogen bond, and it is an intermolecular bond. However, it's a very strong intermolecular bond. Okay, so the oxygen in this case, which has a partial negative charge and a lone pair of electrons, this is functioning as the hydrogen bond acceptor. Okay, and there are other chemical species which can be hydrogen bond acceptors other than just oxygen. The two characteristic features that you need to have in order to be a hydrogen bond acceptor is you need to be partially negatively charged, and you also need this lone pair of electrons here. Okay, both of these are then going to interact with the partially positively charged hydrogen. Okay, the hydrogen bond donor. Right, so we are now going to form hydrogen bonds between this hydrogen here, which is a hydrogen bond acceptor. Okay, so it's got a partial positive charge. And this oxygen here, which is a hydrogen bond acceptor here. Sorry, that's the hydrogen bond donor, and this is the hydrogen bond acceptor. Okay, so that's the scenario that we've modelled here. So we're going to form a hydrogen bond between that hydrogen and that oxygen there. Another one we're going to form is between this nitrogen here, which will have a lone pair of electrons. Nitrogen has a greater electronegativity than carbon, so it will also have a partial negative charge. The hydrogen here will have a partial positive charge because it's again attached to a nitrogen, which has a far greater electronegativity than itself, so we can form a hydrogen bond there. And then again, we've got the identical scenario to up here, down here, so again we can form a hydrogen bond there. So we can form these 3 hydrogen bonds between cytosine and guanine organic bases. Okay, so this is a cytosine-guanine organic base pair. Okay, right. Now let's talk about the groups that are available for transcription factors to bind on either side. Okay, so remember, transcription factors again bind by hydrogen bonds and also by hydrophobic interactions. Okay, so they can interact with groups that are hydrophobic. Okay, so let me just talk about what a hydrophobic group is. We're not actually going to see one here, but we will see one when we look at adenine bound to thymine. Okay, so what's a hydrophobic group? Well, hydrophobic group is a group that tends to avoid water, okay? And the reason it avoids water is that it has an incredibly neutral structure. Now, why does this mean that it's not going to interact well with water? Well, because water is so polar. So let's draw water here. Okay, so here is our water molecule. Now, oxygen has a much greater electronegativity than hydrogen, so in both of these bonds, the electrons are going to end up sitting closer to the oxygen, giving the oxygen atom a partial negative charge. Okay, so the oxygen is going to get a partial negative charge. Meanwhile, that will leave the hydrogen atoms with a partial positive charge. Okay, like so. Right, so this means that water can interact brilliantly with other water molecules because you have a hydrogen bond a hydrogen bond acceptor up here, and you have hydrogen bond donors down here. Okay, so if we put another water molecule here, okay, this oxygen will have 2 lone pairs of electrons, it's got a partial negative charge, it's got everything it needs to be a hydrogen bond acceptor here. We've then got a hydrogen bound to a far more electronegative atom, oxygen, which therefore has a partial positive charge. It is therefore in a perfect position to act as a hydrogen bond donor, so we can form a hydrogen bond in between hydrogen atoms and oxygen atoms. Okay, so water molecules combine to each other by very strong interactions. If, on the other hand, you bring in a hydrophobic molecule, which is extremely neutral in structure, okay, if you take a very neutral molecule, and I'll give you an example so that I have something to point at. Okay, so an example of an extremely hydrophobic molecule would be something like stearic acid. Okay, now stearic acid is the old name for a molecule that would now be called octadecanoic acid. Okay, and although octadecanoic acid is a bit more of a mouthful than stearic acid, it's a useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with an 18-carbon, fully saturated carboxylic acid. Okay, right, so here is the carboxylic acid group. Then we need 17 more carbons after this first one. So 16 of these are going to be in the form of methylene groups. Okay, here's a methylene group, and we're going to repeat it 16 times. And then right on the end, after those 16 methylene groups, we're then going to have a methyl group. Okay, so this is octadecanoic acid now. 1, 2, and then 16, that takes up to 18 carbons. Okay, right, so this extremely long, fully saturated tail, this is extremely hydrophobic. And the reason is it's extremely neutral. So why is it extremely neutral? Well, it consists of bonds between carbon atoms. Okay, now carbon has the same electronegativity as carbon, so of course the electrons are going to sit in the middle of this sort of a bond. In addition, we've got bonds between carbon and hydrogen atoms, and carbon and hydrogen roughly have the same electronegativity. Okay, so the electrons will once again sit in between those two. So none of these bonds are going to be polar, so everything is just going to be neutral basically in this structure, which means that if you ram a water molecule against this, it can't interact that well with this sort of a molecule. Okay, so if you try and mix a fat sort of molecule, which is a hydrophobic molecule, okay, so that's the characteristic feature, by the way, of fat molecules or lipid molecules, that they have an extremely neutral structure and are therefore hydrophobic. If you try and mix fat with water, what will happen is they will separate. The two layers will always separate if you leave it alone. I mean, you can shake it up and get them to mix for a while, but of course they will separate if you then leave it alone. And the reason is that the water molecules just don't want to interact with the fat molecules, okay? So they try and adopt a conformation of the system where the interaction between water molecules and fat molecules is minimalized. And the way that you can achieve this is by separating the two layers. So you aggregate all the fat molecules together, you shove them all into a corner effectively, so that the water molecules don't have to interact with them, so that the water molecules can all interact with each other. Okay? So by separating the two layers, it results in this interaction between the fat molecules and the water molecules being as small as possible. And clearly it will depend on what type of fat we're talking about as to whether it takes the top layer or the bottom layer. Okay, but the principle is that they will separate always. And it's not because the fat molecules like to interact with each other. Okay, they interact in fact really weakly. It's just that water despises interacting with them. Okay, so it shoves them all together so it doesn't have to interact with them basically. Okay, so that's what a hydrophobic interaction is basically. It's where when neutral structures are aggregated together so that water doesn't have to interact with them. Okay, right, and we're going to see that transcription factors sometimes bind to DNA via hydrophobic interactions. But it's mainly by hydrogen bonding. Okay, so let's have a look at the groups that are left over on this cytosine-guanine organic base pair that are remaining and can form hydrogen bonds. Okay, so we'll highlight hydrogen bond donors in red and hydrogen bond acceptors in vivid purple. Okay, right, so here is a very obvious hydrogen bond donor that is free. Okay, and that's on the major groove. So remember this side where we've got the smaller aspect, this is going to be the minor groove of the DNA, and this larger aspect is going to be the major groove of the DNA. Then also facing the major groove we have a hydrogen bond acceptor there, because remember that oxygen is going to have another lone pair of electrons there, and it's also partially negatively charged. And we also have a hydrogen bond acceptor here, because that nitrogen there is going to have a lone pair of electrons here as well. On the minor groove, we have a hydrogen bond acceptor here, and then we have another obvious hydrogen bond donor, which is here. Okay, right. We will come back to this picture, because what we want to see is that when we look at the other organic base pair, which is adenine paired with thymine, that on the minor groove it's going to have pretty much the same availability of hydrogen bond interaction sites as for cytosine and guanine, but we're going to see that on the major groove it has a very different pattern basically, and this is why transcription factors which want to recognize specific sequences of organic bases need to bind in the major groove of the DNA. Okay, right, so let's now draw the other organic base pair. So we're going to do thymine now bound to adenine. Okay, so we'll start off with thymine. Okay, so once again we're going to have rotated the structure a little bit, but I'll try and draw analogies as much as possible. Okay, right, so this is going to be our pyrimidine ring here. Here is one of our nitrogens, and here then up here is another nitrogen there. Right, okay, then off this carbon you're going to have a double bond to an oxygen atom. Okay, so if I go back and show you where I'm working from, here is thymine. This is the nitrogen which is going to attach to the ribose sugar. This is this carbon with the oxygen coming off that's in between the two nitrogens. Okay, right. Then coming off this nitrogen, we're going to have a hydrogen coming off there. And then coming off this carbon, we're going to have a double bond to an oxygen. This nitrogen then will be bound to the ribose sugar, or rather, sorry, the deoxyribose sugar. We're talking about DNA here, so it will be a deoxyribose sugar here. Okay, right. Then we have one further thing to put on here. Okay, we've also got that methyl group right at the back, so that's going to be coming off up here, and this is why I've talked so much about hydrophobic groups, because that methyl group is going to be a little glimmer of hydrophobicity on the major groove over there. So this is our thymine organic base here. Okay, Now let's put our our adenine organic base next to it. Okay, right. So once again, this one will be a little bit easier to draw the parallels for. Okay, so we're going to have our pyrimidine ring here. Okay, and this one once again is just going to be on a slant, basically. Okay, and remember the other nitrogen of the pyrimidine ring will then be down here. Okay, you've then got a double bond here, a double bond here, and a double bond here. Okay, then in the case of adenine, you then have an amino group coming off here. Okay, so remember you have the intact pyrimidine ring, and all that you've then added onto it is that amino group. Then we need to put the imidazole ring in here. So here are these two nitrogens, here's the imide bond here, and then here's a single bond from that carbon to this nitrogen down here. And then we're going to attach this nitrogen onto a deoxyribose sugar over here. Okay, right, so there is our adenine organic base here. Right, so now let's see the hydrogen bonds that form between the adenine and the thymine organic base. Well, once again, we've got a hydrogen bond acceptor here, a hydrogen bond donor here. Okay, so we're going to get a hydrogen bond forming there between that oxygen and that hydrogen. Then we've got a hydrogen bond donor here and a hydrogen bond acceptor here, so we'll get a hydrogen bond forming there. But in the case of adenine and thymine, I pointed them in the wrong direction. You only have two hydrogen bonds forming between those two organic bases, whereas between cytosine and guanine, you have the three hydrogen bonds forming between those two organic bases. Okay, right. Now what we want to talk about is the big topic, which is what hydrogen bonding groups are available on the major groove this side, and what groups are available on the minor groove this side. Okay, so once again, we'll denote hydrogen bond donors in red and hydrogen bond acceptors in vivid purple. So, hydrogen bond acceptors. On the major groove, we have this as a hydrogen bond acceptor here in red. Okay, we also have this oxygen because that has another lone pair of electrons that isn't involved in this hydrogen bond. Those are both hydrogen bond acceptors. Then we have in the middle a hydrogen bond donor there. We also have this hydrophobic group here, which is available for hydrophobic interactions. Okay, then on the minor groove here, we have these two hydrogen bond acceptors here, this nitrogen and this oxygen. And by the way, I've just realized that I have forgotten to mention that we had a hydrogen bond acceptor here on the cytosine and the guanine organic base pair. That nitrogen here was a hydrogen bond acceptor as well. Now, if we draw the parallels here, these two hydrogen bond acceptors here on the thymine and adenine, those were the same on the cytosine and guanine. Okay, we've lost this hydrogen bond donor here, but that's all that's changed here. Okay, so they've got very similar minor groove appearances. Whereas if we now look at their major groove, we had a hydrogen bond acceptor, and a hydrogen bond donor in the case of cytosine-guanine organic base pairs, whereas we now have a hydrogen bond acceptor, a hydrogen bond donor, a hydrogen bond acceptor, and then a hydrophobic group. Okay, so this is the main reason that transcription factors bind always to the major groove, because that's where they can more easily distinguish the different types of organic bases, basically. Okay, and therefore they can have specific sequences of organic bases which they bind to. Okay, right, so that's a very long-winded explanation of why transcription factors bind to the major groove of DNA, but I hope you do at least now understand what the minor and major groove of the DNA are, and why they exist, and why they're significant for transcription factor binding. In the next video, what we're going to turn our attention to is looking at specific motifs that are present within many different examples of transcription factors, and what their structures are and how they bind to the DNA.